So with that, I'll start sharing my screen. Here we go. So I hope you've seen in the workshop description that you can get yourself a free account on webknossos.org, and that could be quite helpful for this workshop, because then you can follow along and do stuff as I show it. If you want to do it later on, that's also fine. So once you have an account and you're logged in, this is one of the screens that you will probably interact with the most. It's the dataset list. I now already have a couple of datasets here that I use for demo purposes. So when you open it up for the first time, it's probably empty. But yeah, so the first part of the workshop is basically to show you how you can get some data in there. And there's the Add Dataset button up here. And this allows you to upload data that you might have. So here I have a TIFF, an OMI TIFF. Basically, it's a 3D data set. It's around 1 gigabyte in size. And I can just drag and drop that onto here. And then I can give it a name. So, call it something like this, L4 sample, volume, OMI, whatever. And then I need to specify also the voxel size, because that's currently not automatically read out of these files. When I scroll down a bit, I can see now that the file's been picked up, and I can start to upload. I'm not going to do this right now. I already did this before the session, just to save a bit on time and not to bore you while the loading screen is progressing. So I'll show you what that looks like. It's basically this data set that I have here, and yeah, once the conversion is done, you will also, or the upload is done, you will also get an email, and then you can just click on that link in the email, and then you will end up in this screen right here. So this is the main view of Webknossos, where you can interact with the data. You can browse around, of course. You can use a visualization tool with your mouse, just dragging and dragging around. That works in all 3 dimensions, and the data will be loaded. Basically, if you move around, then the data will be loaded in just to fill up your screen. On the right-hand side, you can see some of useful shortcuts. So if you want to zoom in, that's I, and zooming out is O. It's with some touchpads, it's also like the pinch to zoom works. Also, if you want to work with an iPad or something like that, also you can do pinch to zoom just to zoom in and out. On the left-hand side, there's a sidebar where you see a histogram of the image that you uploaded. And so this is an electron microscopy image that we're looking at here. And yeah, I could optimize the histogram a bit manually just to better fit this distribution. But there's also an automated feature that you can just click on clip histogram, and then it will basically clip the histogram for you, so you don't have to do that on your own. If you're working with other modalities, like if you have light microscopy data, then it will probably have multiple of these layers, and then you can also assign colors to them. And then you will be able to distinguish them better, and yeah, start working with that. So that's basically the general way of how you can visualize your data. And one of the main features of Webknossos, of course, is doing annotations on top of that. And to do that, you just click on the button, create annotation, and the view now looks pretty similar. You have 2 more layers now. One is a volume layer that is for the segmentations that we'll create in a second, and the other one is for skeletons, and there are a couple of settings that you can put in here. And also on the right-hand side now, we have more tabs that are useful for the annotations that we're doing. So I think we'll just start with doing some volume annotations, and I'll start just with the basics. So there's a brush tool that you can use to brush around. So I can do this on a couple of sections. You may have noted that I just skipped a few, because Webknossos has a volume interpolation feature, so that you can interpolate in between these sections, and that can save you quite a bit of time when working in 3D. it's a linear interpolation, so you need to check if it matches the quality requirements that you have, but it can definitely speed up the process quite a bit to do that. So this how this works. Over here is a setting to basically change the brush size, and there are also a couple of presets that we just added, and that's quite useful if you want to switch quickly between different brush size, and you can also customize them to your tasks. So that's also possible. And some people even map these to their like if they have a Wacom tablet or something, sometimes you have like buttons on the pen, so you could even map it to that so that you can switch very quickly. So that's the basic way of doing a volume annotation. By the way, I'm moving with my the space key on my keyboard in Z here, Z direction, and when you press Shift and space, then you can go backwards. And there's another very similar tool. It's the trace tool, and it differs from the brush set. It's basically you just do a trace outline, and it fills up the inner part as well. So it's we have these two tools because people have different preferences of what you use, and we want to basically build accommodate both scenarios. So let me just finish this up and interpolate here, and now I've annotated part of this cell here. On the right hand side, you can see there's a segment tab. So I have not one segment at the moment. I can create a new one by clicking this button up top here, or pressing the C shortcut. And then as soon as I draw, maybe let's do this over here, a new segment will appear in the list. And it's a bit hard to see the color here, but that's now here. This segment list over here allows you to organize stuff, so you can give it names. You can create groups, so you can put in these segments into groups if you wanted to, and they can give them names and organize everything. So that's not particularly useful if you only have 2 of them, but if you have hundreds of thousands of them, then that's certainly a more useful way of organizing this. What Webknossos also can do is it can do 3D representations of what you've just drawn. So that's what we call a mesh, and you can then visualize that in the 3D viewport here and see that it's and you can also basically do that for all of the objects that you might have. So I'll do it all in one batch. I didn't draw much here, so it's just this tiny piece at the moment, but that also works. And these will update as you continue to draw. And if for some reason they don't update, then there's also a refresh button. So that's also quite handy to know. And then another thing that you can do is you can download these meshes, and voila, then they appear in your on your file system. Let me just open this up with a viewer. So my Mac has a feature to just visualize them in here, but you could now go and import them into Blender or Amira or whatever your favorite tool is for visualizing these kind of meshes, and then you can use that to make a figure or a video or something like that. So yeah, there's also an eraser tool. It works exactly like the 2 draw tools that we have. So there's a trace eraser, and there's also, oops, I now draw with the wrong color, and I just noticed that. So let me switch back to this one and just draw over just very quickly to correct that. So yeah, but I was just saying, so we have the eraser tool, and it can either work in the trace mode, or if you're in the brush mode, then you can also have a brush eraser. There's another way of switching the active segment ID. There's a pick tool, and if you do that, then that's also the way, and that's indicated in the segment list by this little brush icon right here, which one is the active right now. And also down here, there is a segment ID. So that's might be a bit more advanced to use that. Okay, so these are basically the manual or the like the manual tools of doing that. There's also a semi-automated feature in Webknossos. It works with the segment anything model that has been published by Meta AI earlier this year. And the way it works is you, well, let me quickly go and make a new segment ID. So you open up a bounding box like this. And we generally advise to make the bounding box fairly precise and then let go of it, and then it does it best to fill it up. And this also works in combination with the volume interpolation tool. So right now I did confuse this MIDO here, so let's give it another try and just fill it up here. So that worked quite well. And I'll probably go this far and then stop for now. And then, as I said, it also works well with volume interpolation feature. So this is a very fast way of generating some volume annotations. It's not perfect all the time. That's why it's still useful to have like the brushes around and to fine tune that. So this is the quick select tool. There it has the AI based mode. Also, we have a non-AI based mode. It's just a simple watershed algorithm that can also work quite well for some for some objects. It doesn't work super well for features that have that might have a clear membrane or boundary there, as we're seeing here, but have stuff in the middle like this MIDO here. So that's where the AI feature is much better. Yeah. So are there any questions at this point about the volume annotation features of Webknossos? Right. You can also post them in the chat anytime if you want to. So yeah. Okay, so another feature that Webknossos has is skeleton annotations, and that's something that we implemented first, basically to trace neurons. So a skeleton is, as the icon might suggest, a graph representation. It's like a line segment graph, and you can use that to click on points, and then right here we can already see that this is now growing and going here, and voila, this the neuron is done here. So I'll just switch to another viewport, maybe here, and continue tracing here. And it's more difficult to trace now because there is a MIDO directly in the way, but I'll just follow along here. And maybe I'll now switch back to here. Basically, what we typically do is we look for where is the object around us and then trace in that viewport. And okay, that's the end of the dataset. So, and now what I just created is this short skeleton trace. There's a skeleton tab where, again, you have like this hierarchy that where you can organize everything into, and you can get some so if you have some more features, like you can change the colors of the trees, you can delete it, of course, you can get the length of it. So I just traced almost 4 microns. You can always also get that, or you can, some of these features you can also get by right-clicking on this thing right here. So for example, I could get the position of this node right now, or I could get like the distance to some other place. So for example, if I don't want to have the path length of the full skeleton, but just of a part of it. So like I did just right now, I have this active node over here, and I right click on another node, then it tells me, okay, there's 2.6 microns in between of path length along the skeleton that this is long. So this is useful for, as I said, tracing neurons, but also other tube-like shapes work as well, like vascular systems or something like that. That can also work quite well. So it also has support for branching. I didn't see a branch right now, but let me just go back to here and just show you what that would look like. So you can click on B on your keyboard, and that will create a branch point, and then you can start continue tracing somewhere else. So this is a bit of nonsense right here. Right now you can see that we have created this little branch over here, and you can even so Webknossos keeps a list of these branches. So when you click B multiple times, and you can continue there, and then there's the J key where you can pop back to a branch point and then continue in the other direction. So yeah, that's the basics of the skeleton annotation tool. There are a couple of options you can do, like you can increase the size of these things. You can also increase the clipping distance in the 3 viewports over here. So how much of the new of the skeletons you see. Yeah, and a couple of other options that might be useful. What you can also do is, like, you can give these names again. I think I was saying that already, but you can also attach comments to individual segments or nodes. So do something like this node over here, and then I'll just go over here, and then you can basically easily go back to that. So that's like a bookmarking feature, or it's also useful if you want to collaborate with other people. Then you can leave notes for them, or you can put in some analysis information that you can then even later on automatically evaluate with a script if you want to do that. Okay, so then just for completeness sake, the third type of annotations Webknossos supports is the bounding box tool. So the way it works, you can just make a bounding box and resize that to your liking. And that's useful to just delineate parts of your image. For example, if you want to assign annotation tasks to different people, then you can just create these kind of bounding boxes, give them a name, who should work on it, and then that's a way of organizing that. You can also download these bounding boxes. So I'll just click on this right here, and then this modal will just pop up, and it will allow you to export the images that you've created in the side of this bounding box. So you can select the format. As of today, we support only TIFF and a TIFF stack. And you can pick the layer that you want to have. So layer are the ones that you see on the left-hand side here. Right now we only have the 2. So that's the original data, like the EM data, and the other one is the volume annotation that I created. I don't think there is any segmentation that in this bounding box that I just have, but if you had that, then you would get an ID mask that you can then download and use in other tools. So you can also pick the resolution that you want. So 111 is the highest resolution, but if you have a large bounding box, then you might not want to download it at full resolution. So you can pick whatever suits your need, and there's a small indication of how large the files would be. And there's also a limitation just in terms of how many or how many voxels you can download in one go. Basically as a safeguard to not overload the server, but so it's a gigavoxel, and if you want to, if your bounding box is larger than that, then basically you have to pick a lower resolution so that it still works. So now I could press export and it would do that. Maybe I'll just do that because it's a very, very small piece of data that I just have, and it will basically convert that on the server. And once it's done, then it will start the download process. Actually, the data I was showing you earlier, this is exactly, well, I just downloaded it from Webknossos. So this is another example of what it would look like, And now the other one is also done. So let's try and put that into Fiji. Not an expert on Fiji, but yeah, it also has a volume of like the down sampling pyramid in there. And yeah, but now you can see, I mean, this bounding box is tiny, so there's not a lot of detail to be seen here, but this is what you would get. And as I said, this also works with any annotations that you do, and then you could view them as a segment ID map in another tool. I'm not 100% sure if how that works in Fiji because I don't know too much about Fiji, but I'm sure there would be ways to import it there as well. Okay, and just recently we added a measurement tool, and it's 3, as the name suggests. It's just like you click somewhere and then you can draw a path, and it's similar to the skeleton tools, but it's not persisted. So it's really just, okay, I can do like this ad hoc measurements of how big is this diameter of this object right here. So that would be 0.6 microns. Roughly, and there's also an area tool, so where you can basically just brush around and yeah, and it will give you the area right there. So maybe one thing that I haven't talked about, maybe I just forgot because it's such a new feature. So let me just go back to the segment here. And now I can right click on this segment, and there are also a couple of statistics that I can get here. So I get the volume. For example, I get the bounding box of the shape that I've drawn, and the other one is, well, it's the distance to the active node. So that's not super useful at this point, but these are segment statistics that Webknossos calculates. And there's also a way of getting them again as like in a batch processing kind of way. So now basically I've drawn 3 segments, and here are all of the statistics I can get that, export that as a CSV, and then you can load that into Excel or into a Jupyter notebook with pandas or something, and then start doing analysis on top of that. And that also works. So if you just want to get that for a single group or so, that also works. So that's why the grouping feature it can be quite useful. Yeah. Okay. Are there any questions so far? No? Okay. Well, just keep going. Next on my list is well, already a bit more advanced feature. So Webknossos is well, it's a server-based tool, and one of the ways you can make use of the property that is a server is that you can basically access that data also from a programmatic setting. Like here I have a Jupyter notebook, and there is a Webknossos library that you can just install with pip, as you would always like a regular library. I already did that just to save a bit of time, and then it will do that. And then here you can see we are importing this library for Webknossos as WK, and then we can work with that. And the library basically gives you access to all of the kinds of data that you might have in Webknossos. These examples start with local data sets, so you could prepare your own data set locally in Webknossos and then upload it later. So I'll just walk you through the code very quickly here. I already executed, as you can see. So here we are creating a new data set. We're giving it a file path where it can store it on disk, and we're giving it a voxel size in nanometers. So it's 4430 right here. And then we can add a layer. So the layers are, again, these things you see on the left-hand side here. We're adding one of them, we're calling color, and we tell Webknossos that this is a type color category. There are 2 types. One is color, and the other one is segmentation, and that will basically change how the Webknossos UI gives you access to the tools. Like the segmentation tools are only available for segmentation layers, for example. And then I can also specify the data type, which happens to be 8-bit unsigned. So now I have a layer, which is great, but to actually be able to write some data into that, I also need to add a MAC. So a MAC is what we basically call a resolution level. And 1 is always the highest resolution you can get. So I'm creating this object. Now I'm just randomly generating some data here. That's just a toy example, but here you could load in some data that you get from a TIFF file or something, or from that you may have an HJ5 or some whatever kind of NumPy array basically you have in Python. And then you can write it out and tell the library where to write it. So right now I'm just writing it at the beginning of the data set. So we'll be up in the top left corner then. So now it's written onto disk. Now you could go about compressing that data. That will save a bit of space on your hard disk. This has been particularly helpful for segmentations because Webknossos stores them as ID maps, and that's highly compressible. So you can really shrink down data sets by factors of, I don't know, 10 to 100, I guess, somewhere in that range. And then the next function that we're calling here is downsampling. And what this will create is the resolution pyramid. And it will take into account the voxel size so that it will do some, can keep this anisotropicness in mind while downsampling. So yeah, it will do that. And there heuristic when it will stop downsampling. So now it figured out that from the size of the data set, max 16 is enough. So that's where it stops. So this is now a local data set, and I could now call dataset.upload, and then it would upload that to your account. I won't do that right now because for that I actually need to authenticate myself, and I can show you in a second how that works. Cool. So, but another thing that you can do is working with datasets that are already on Webknossos. That's what we call remote datasets, and it's actually quite easy. So you just do wk.dataset.openRemote, and you paste in the URL that you have over here. So this is now an annotation. So if I go back to just viewing that, it should also work. And I can basically just take this and yeah, put it in here, and then we are able to access it. Again, because I haven't authenticated myself in the Python library yet, I'll stick with this public data set that we have here, and just show you how it works. So again, I get a dataset object. I can query the layers that it has, and it has 2 layers. So it's this color and the segmentation layer. Maybe we'll just open this in a separate tab so you can see what this looks like. So this is what this data set looks like. So it's the same EM data we've been looking at, but now it has a segmentation from an automated segmentation that we've done. And yeah, this is what it looks like. So here we see the 2 layers again, the color layer and the segmentation layer. Color is the EM data. And then I can access the resolution level or the MAC again. There's a feature called get finest MAC, which almost always gives you MAC 1, but if in some cases you might not have MAC 1, and then it will give you the best resolution basically. Yeah. And so now next in this example, what we'll do is we'll try and basically make a local copy of this data set. So for that I create a data set, an output a copy. This 2Python has this nice file viewer here. So you basically get this just an empty folder with a JSON file in it. And yeah, and now I'll prepare this data set. So I'll create a new layer for that called color copy. And this is the out data set where I add this. And now from the data set that the remote data set, I'll pick the color layer, I'll get the bounding box, copy it over, and then also create a MAC. So this is just a bit of setting things up. I'll do that real quick. So that's done. And now up for the actual copying of things. So there are some features in the library that allow you to do chunked operations, and that can run in parallel. For zipped chunks is one of them, and it's quite useful for copying because you can give it 2 data sets at the same time, and then it will do that. So yeah, I'll just run this right now. We'll see. I haven't tested it, so I don't know how long it will take, but you get a progress bar. It probably will give us just 50% and 100%. So that's we'll know. So yeah, this is obviously a bit advanced use of what you can do with Webknossos, but if you're doing if you're into coding, then this library can be quite useful to work with Webknossos. And I'll just show you a bit what's also going on in this notebook here. So another thing that you can do is use a custom executor. By default, it will just use multiprocessing, so local on that machine. But you can also use a Slurm executor, for example. So if you have an HPC cluster at your institution, then you could use that and spawn jobs to that, and then it will run highly parallelized. So that's useful for very large data sets. Yeah, okay. And another feature that could be useful is generating Webknossos datasets. So in the earlier example, I showed you how to write out random data that which is not super useful. So it might be more useful to actually convert some data that you already have. And for that, we have a function called add layer from images. And what you can do is you can give it like a file path or with a wildcard in it, for example. So this would be a TIFF sequence, and it will create a new layer for you automatically. And this works with a bunch of different file formats. Basically, all of the file formats that are also supported with the upload feature in the browser version. And it will create that. Then again, you can compress and downsample that, and then you could be able to upload that. All right, so this is still running. It's probably going to take another minute or two. Yeah, so it now created this folder for this layer, created a folder for the magnification, and now it's writing in all the data. So yeah, I don't know, since I'm just giving a bit of an overview of what kind of features there are, I would leave it at that. If you have any specific questions about the Python library, I'm of course happy to answer them. But I also want to point out that we have a documentation on that you can find at docs.webknossos.org. And if you go to the Python library tab, there are a bunch of examples of what you can do with all of this, and there's also an API reference that lists out every single function that there is, or so what you can do. And yeah, if you can read on that on your own, and if you have any questions, then you can reach out. Maybe that's a good thing to mention. I'm sure most of you know about the image.sc forum. Webknossos is also present there. So if you go to community partners and click on Webknossos, then you can ask any question here, open a new topic, and then we'll be happy to help, or somebody else from the community will also be happy to help. All right. So let's just check. So the copy is done now, and yeah, that's great. It still shows 50%, which is probably just a bug in the visualization. So it shows us the tick here, so that's great. Okay, so the next feature I want to talk about is working with Zarr images. So if you don't know about Zarr yet, Zarr is a next generation file format that a bunch of different tools support, and Webknossos also supports Zarr. And one of the nice things is that you can load data into Webknossos without having to upload that. So if you already have data stored somewhere else, like in an image archive, then you're able to import that data into Webknossos without basically needing to upload that, so it can be streamed remotely. And well, Webknossos technically also supports other formats like NeuroGlancer, precomputed, and N5. So if you have any of these data sets, that should also work. So okay, so what I just did is I clicked on Add Dataset, and you have the second tab, which is called Remote Dataset, and it means remote because it's not going to be uploaded, but it's going to just be linked via an URL. And now we can pick a data set. So one place of where you can find some data is the IDR, Zarr gallery. So they have a catalog from the IDR is from the folks at Omiro. And yeah, we can just pick a data set. So let's maybe pick this one. So if I click on this, I have to click here, then you get this Zarr. So it's another visualization tool that's web-based. And so I had this one. This time I'll just copy the URL, paste it in here. And what we'll do, we'll check if everything works. Like I need to give it a name. So I'll just use the ID for now. this, and it will figure out most of the metadata on its own. So figure out the voxel size. These numbers look a bit weird because Webknossos currently assumes that it's in nanometers, and then numbers can get a bit big and yeah, and long, and it will figure out all of the other stuff. So yeah, let's just do that. I'll click import, and then I can open up this data set. And so now I'm already seeing the segmentation to also be able to see the well, fluorescence data. I'll have to press clip histogram to see it, right? So now here you can see it. And yeah, and most of the features that I have been showing so far also work with this remotely stored data. So like all the annotation tools, including the segment anything, works as well. Can pick different colors if for any reason you wanted that. Yeah, so that's the way how you can import Zarr data into Webknossos. It's pretty as simple as just putting in the URL and pressing a few buttons. But another feature that I wanted to show you is that you can also go the other way. So get data from Webknossos out as a Zarr. And for that, I will, let's go with this data set. So I'll go to the menu here and I'll click on Zarr links, and I can create a new Zarr link. And I have to do this because it's a private data set. If it were a public data set, then there would already be a standard link already. So this is another tool, NeuroGlancer, developed by Google, and just want to show you how that could work. So if you copy this URL that Webknossos provides you, and you put it into another tool, and voila, the data is showing up. And this works for the image data that we uploaded. As you remember, we uploaded this data, but it also works for annotations that you create inside of Webknossos. So far, it only works for segmentations. Yeah, and now I need to find the location where I actually had something. So let's just copy this over. And voila, this is now I can also see these segments in NeuroGlancer as well. And this works with a bunch of other tools as well, like with Zarr. Well, I think there might be a bug in Zarr where it doesn't work, but that probably gets fixed soon. And there's also ongoing work to make this work in Fiji as well, so that you can basically have your data inside of Webknossos and then access it from Fiji or NeuroGlancer or another tool. So that's a useful way of using Webknossos as a data management system. Even if you don't, or in combination with the annotation features, but you can even use it without the annotation feature of Webknossos, just use it as an image database. All right. So that's Zarr streaming. Again, if you have any questions, feel free to just unmute yourself or write something in the chat. Or if you have any questions, you can ask them right now. Okay. So is the next couple of minutes, I want to talk about some of the upcoming features that we're working on. So some of these demos might not work in the first run, so please bear with me. But one of the features we're working on is adding a correlative multi-modal features to Webknossos, so that you can map or register two data sets into one scene in Webknossos. And this is what the output of that could look like. So here already we actually used a big warp to click landmarks, and then we could import them into Webknossos. And these are so one of the data sets is warped, and you can see which one by this little icon right here. So this one is the warped one, and this one is the one that basically is the original coordinate system. you can do. And now I can toggle this, so I can also switch it over. So now we have the blue one is basically in its original form, and the green one is transformed. And this toggling is useful because then you can do the annotations in whatever basically is best for that, for that kind of for the data that you have. And so by the way, what we're looking at here are two X-ray, so micro CT data sets, and one has been taken with a lab X-ray system, and the other one is taken at a synchrotron by our collaborators at the Francis Crick Institute. So yeah, this is a feature that we're building at the moment, and we're probably in one of the next, or maybe in the release next month, we'll add a feature so that you can import the landmarks from Big Warp, but also will offer a flow so that you can click landmarks inside of Webknossos to then register these data sets, and then have then a registered data set that you can then use for doing annotations. So, and if you have any feedback or any ideas what we should include into these features, then I'd be very happy to learn about that. Okay, so there's a question in the chat about does Webknossos work with DICOM images? So that's a good question, and I don't know from the top of my head. Webknossos, when you upload data, then it gets converted, and I'm not 100% sure if we have a DICOM converter for that at the moment, but it doesn't have native support for DICOM, so there's it would require a conversion step in that. Yeah. Okay. Good. Yeah. So as I was saying, so that's transformations, working on that, and we're happy to get feedback. If that's useful to you, then please let us know what kind of features you would need. All right. Another feature that we recently added, but it's not fully supported yet, is a time series support. So it's not fully supported. So one of the limitations, for example, is that we don't support uploading yet. But if you do happen to have a data set that is in Zarr which has time series data, then that should work. So I haven't tried it with this one, but let's see if this works. And so upload is coming in. It doesn't work with this. Okay, let's try another one. Upload is coming in one of the next releases, so probably before the end of the year we'll also have upload support for time series data as well. So okay, I'm just now randomly trying out some data sets from this list here. All right, so it seems to be picked up. Let's give it a name and let's import it. Fast loads. Let's just stick to one channel for now, just so that things load a bit faster. So here you can see now, well, again, it's a 3D view, right? But the new thing is that we now have this additional UI element up here where you can select the different time points, and it will then load in the data for that particular time point. And the annotation features also work with the time series data. So you can now start drawing or creating skeletons on top of that. So all of that still or also works for time series data now. And yeah, as I said, it's a very new feature, so there might be some bugs here and there. If you find any, then please let us know in the forum or also on GitHub. And we're looking forward to basically ironing out all these bugs so that it becomes useful. So that's time series support. And then the last feature that I wanted to show you is also, oops. Sorry. Oh, well, one useful feature is that there's also an undo feature. So if you happen to brush or do something dumb, then just does that. By the way, it also has an autosave. So if you you don't have to, it saves every 30 seconds or so, so you don't have to click this button on your own, and everything will be stored on a server and back up there, so you don't have to worry about that kind of stuff. Okay, but what I wanted to show you now is automated segmentation, and that's a very early preview, and for the moment only works for these kind of EM brain data, and even not for all kind of microscopes. So it's highly experimental at this point, but I still want to show you how it works. So again, it works by you creating a bounding box first. So let's just do that. And then there's in the menu, there's AI segmentation, AI neuron segmentation, and you get this model. And then you have to select the bounding box. So let's go with this one. This is the one I just created. And I can press start AI segmentation, and then it will run a job on the server. And that might take a while because it's a full deep learning model. And depending on what kind of size you feed into it, that might take a few minutes, or maybe some cases even hours to process. And for now, once while we're still rolling this feature out, it's also limited in what kind of sizes you can put in. So it's really meant as a preview at this stage. So just to save time, again, I'm not going to click on this, but I'll show you, like, again, in the public data set, what this would look like. And here we actually have the full data set that we had, and this is the segmentation that has been created with that feature. And another feature that I can show you is that we also have pre-computed meshes, and this is helpful when you have a very large automated segmentation. Then you can also generate these meshes, and they will load instantly. Whereas the meshing I showed you earlier is generated on the fly, which is great when you're annotating, because then you can incorporate that data. But it's fairly slow for large objects to render out meshes. Yeah, so that's a feature that if you're interested in, you should keep an eye out over the next, let's say, year or so, because we're rolling this out and we're figuring out the infrastructure and figuring out what kind of models that we want to include with this. So mitochondria models are next on our list for EM data, and then we're also thinking about integrating models from the bioimage model zoo. So that basically it becomes very easy to use these automated segmentation models from Webknossos. Okay, so there's another question in the chat. So we do not have support for high content screening at the moment, and it's not really on our roadmap for the next year, I would say, because we want to focus on these AI features and the transformations. But if we get more demand for that and more people ask about it, then we'll put it on the roadmap, and then it might come at a later point in time. Yeah, but thank you for the question. Okay, so that were all of the topics I wanted to cover today as part of this overview. If you do have any questions, then we can do now a bit of a Q&A session. So for that, feel free to just unmute yourself and ask a question.
Hi. It's maybe a bit of a weird question, but is there or could there be a same like group or hierarchy system for the bounding boxes as for the segments? So if I want to make a group of bounding boxes separate from one another.
Yeah, that's actually a good idea. So we got some feedback from people that it's a bit weird that skeleton and segments are in different groups or like in different tabs, and it could be useful to put them all in the same hierarchy, so that, for example, if you draw a neuron with brush tools and then also want to create a skeleton trace of that, then it would be useful to have them like in the same section. And reasons why you would want to do that is, for example, to get the path length of the neuron, or to put in some comments or something like that. So that there are useful use cases to do that. So that's something that we're considering, and then naturally would also just fit to also include the bounding boxes into that. So so at the moment the answer is no, we don't have a feature like that, but I'll definitely take that as feedback, and we'll see if we can put that on the development roadmap. Thanks. Any other questions or anything else that you'd like me to show? All right. Well, then we're almost at the full hour. So thanks for coming. Again, if feel free to try out Webknossos. So we have free accounts on webknossos.org. They're basically have all of the features, and they're just limited in terms of the data that you can upload, because storage is not free. But if you have your data somewhere else as Zarr, then you can load it, and that won't count against your quota. So that also it can work. Webknossos, of course, is an open source tool, so you can also install it on your own servers if you wanted to. And as I was saying, we have the documentation at docs.webknossos.org, also not only for the Python library, but also for Webknossos itself. So if you want to read more about volume annotation tools, you can go there. We also have some videos in there that we've been updating over the last year to basically just give you a head start into how to use these kind of features. But now that you heard me talk about this for an hour, I think you know most of the stuff already. But if you want to know any of these details, then the documentation is a good place to start. And then again, the image.sc is the best place to ask any questions. And yeah, if you find bugs, then you can also put them on the GitHub repository, but you can also just put them in image.sc, and then we'll hope to get to fix them as soon as possible. All right. So thanks, everybody, and have a good day or good night. Bye-bye.